depressed, feel like chronic depression, your telomeres can shorten. Oxidative stress, which is you have too much reactive oxygen in your blood or in your system, and there's not enough of whatever it's wanting to react with it. So you just got these reactive oxygens floating around, reacting with the cell, damaging it in different ways, and that can damage the DNA or the cell itself, causing it to need to replicate more. And exercise can actually, it's been shown to offset the depression and psychological stress, what they do to the DNA or to the telomeres. It can actually offset that. And telomerase. So telomerase actually reverses the effects of telomere shortening by adding the TT-AGGG repeats onto the end. It's only present in the reproductive cells and the fetus before we're born. And so it's repairing our telomeres the whole time before we're born. Then we're born, and the gene stays in all of our cells, but it just shuts off, except for the reproductive cells, it stays on. And this is kind of like if telomerase were a big factor, it would probably look something like this. It's just got the RNA template here, and the end of the strand comes in and it just punches on more repeats. And here's the end of that video I was showing you earlier.
The ends of linear chromosomes.
A little bit more about telomeres.
Important during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats, called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction, and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
Alright, so you guys kind of get what's going on there. Just goes, reads the end of it, adds more repeats onto the end. So this is a graph of mice, the average lifespan of mice, and this is a regular mouse. There's nothing done to it. It's a regular mouse. This is the median lifespan for all of them right here. So it's about just under 30 months. And the G1, G2, and G3, the generations, the first generation of the mouse was completely deprived of any telomerase at all. And it was obviously had a little bit shorter lifespan. And then they didn't do anything more to the mouse. They just, the next generation they tested again, even shorter. The next generation, even shorter than that. So it just gets passed on down through the genes. So what can telomerase do for us? So the experiments were done to determine the capacity of telomerase to reverse or halt the effects of aging at the cellular level. Basically, does it make our cells not age or reverse the effects of aging that's already happened? So the first mouse, they took all the telomerase out, that's the graph you just saw, and it resulted in premature aging and less cancer when compared to a normal mouse. Mouse 2 is a constant overexpression of telomerase in the epithelia, which is a tissue that lines pretty much everything in our body, digestive tract, skin, pretty much everywhere. And that resulted in less aging, but more cancer, which is due to the telomerase keeping those cells active and going. Even if the DNA is damaged, it will just keep adding the repeats on, keep the cell alive and replicating, just keep making more damaged cells. And the third mouse, they called it the super end mouse. They had the constant overexpression of telomerase, but they also added tumor suppressors P53, P16, and P19 ARF. That resulted in less aging and less tumors. Good all around. This is kind of a little picture to show you what's going on. This is the mouse with no telomerase, premature aging, less cancer. He's really old, but he has no cancer, so he's kind of happy, I guess. In the middle, we got the mouse with the telomeres, but no tumor suppressors. So he's nice and young, but he's got a big old tumor right there. And the super M mouse was given the cancer suppressors and the telomerase, and he lived a long, happy life with no tumors or nothing. So how do tumor suppressors work? The P53 protein activates DNA repair proteins, so it activates other proteins that will go in and repair the DNA. And then while that's going on, it will also pause between the G1 and S phase of cellular growth to allow time for the repairs. It doesn't just keep growing and keep growing. It's already replicating before the repairs are done, you know? So the rest is cell growth. And if the DNA is damaged beyond repair, then it'll trigger apoptosis, which is just cell death, to kill the cell. And because of these kind of protective attributes, it was dubbed the guardian angel protein. This is a computer rendering of P53 interacting with a segment of DNA. So P16, it's released when P53 is done and just kickstarts it. Well, it inhibits the retinoblastoma protein by phosphorylation, which means it just adds a PO4 group to the retinoblastoma protein, making it so it can't react with whatever it wants to react with, or it's supposed to react with. And that allows entry to the S phase, because the retinoblastoma protein is what was blocking it from entering before. And that promotes cell growth, obviously. And P19 ARF, which is pretty much there just to protect the P53. MDM2 will bind to P53 and shuttle it out of the nucleus so that it can no longer react with the DNA and fix it up. So P19 ARF is added so that it will bind to the MDM2 instead of the P53 so that MDM2 can't bind to the P53. So in essence, the P53 is like the brake. It slows down the process, gets the repairs going. P16 is the gas. It's assuming that the cell is a car and you're the person in your nucleus or something. So P16 is the gas. Once all those repairs are done, it gets it going again. Does cellular growth. And the P19 ARF is like a backing plate, which is the part of the car which holds the brake onto the car. So it just secures it in place, makes sure it doesn't go anywhere, which is what P19 does for P53. And this is P19 ARF, a computer rendering of the protein. And here, a little confusing at first maybe, but we have experiments done with, they wanted to show how P53 helps, but how the other two proteins help P53 help, you know? So by itself, P53 caused a 9% increase in the average lifespan when compared to, or P53 with telomeres caused a 9% increase in average lifespan when compared to just P53. And the next one, they just added this line here is the P53, P16, and P19. And the next line is those three plus telomeres, and they had a 26% increase. So telomeres is like the key factors, kind of what they're pointing out here as well. And then in the last one, they did P53 compared to P53, P16, P19, and telomeres. There was a 40.2% increase in the average lifespan, which is pretty hefty. So basically this is just a regular mouse, almost. It's got the extra DNA repairs in it, but this is the pairs with all the help and the telomeres that helped a lot. These are the same graphs, but this is the overall survival. This is the overall survival of cancer-free mice. So there's a lot bigger jumps here. 18%, which is double that, 38%, 50%. It's just these proteins, without these proteins, you'd have lots of tumors, is what these graphs are saying. So basically the findings that they had from this experiment was when telomerase is coupled with tumor suppressors, it's been shown to slow the aging process by prolonging the life of cells through telomere lengthening, as well as lessen the incidence of tumors. I'm done.